The title of my presentation today is Chasing Questions, Finding Belonging. And for the rest of the presentation, I am going to be using my voice, speaking English, and that way I will be able to look at my notes as well. I'll turn things over to the interpreter to translate everything into ASL.
So before I can tell you about the sort of questions that guided my journey, I want to tell you a little bit about my baseline. And as any good scientist knows, you need to have your baseline or your starting point to understand the meaning behind your outcomes. I like to think of my journey as being one big experiment, and I think it makes it more fun to think of it like that. And so I was adopted when I was a baby from Huanggang, China. That's this little dot right here. And I was brought to Minnesota, which is this state right up here bordering Canada. And I grew up about 10 minutes away from Minneapolis. It's known for being pretty cold, but I think the summers are the best. They're really beautiful, and they hardly ever get over 85 degrees, which is great and perfect. So a little bit more about the baseline. Here is a picture of me from the orphanage in China. I was wearing about 10 sweaters at the time. The orphanage did this so that the babies looked healthy, but the pictures to send to the parents. So that's why I look pretty plump. This is an older picture of my family from when I was, I think, in 4th grade. My sisters and I were all adopted from China by my mother. I am the middle child. My entire family is hearing and primarily communicates through speaking. My mother first realized I was hard of hearing when I was 2, and she would call my name and I wouldn't respond. So she brought me to the audiologist, and that's when they confirmed her suspicions. They're here. I'm getting ready for my first day of preschool. I hadn't realized until I started making this presentation, but the preschool I went to was actually almost my first introduction to what RIT/NTID would be like, because I spent half my week with other deaf hard of hearing kids and the other half of my week in the hearing classes. After I completed preschool, I was completely mainstreamed. Like many other hard of hearing kids who are mainstreamed, I used hearing aids and I used FM systems. Here's the type of mic I would give to the teachers. And I also went to speech therapy and had a teacher for the deaf. However, as I'm sure many of you can probably relate to, when I was a kid and I didn't understand anything that was going on, I had trouble engaging in classes. I would get distracted or bored, especially when I didn't understand what was happening. My teacher for the deaf at that time realized what was going on, and she ended up asking this question: what if Anna had an interpreter? And this question served as a catalyst. So 3rd grade is when I got an interpreter for the first time, and it became this language explosion that I was super excited about. And I created this schematic or this little cartoon with ChatGPT. And it's pretty cool how ChatGPT can now create things like that. So between picking up sign language and understanding what was happening in class and being able to put captions on the TV, I suddenly had so much more access to school and extracurriculars. It wasn't perfect, but it was a huge improvement from before, which was essentially no access. And then as I grew up and continued to use sign language, I wanted to understand more about the community built around it. So I started wondering what it meant to be deaf. So during the summers, when I wasn't in mainstream school, I looked for opportunities to answer this question. I like to describe these as the summers I learned about deaf, capital D, culture. So if you recognize the title slide, it's from the TV show called The Summer I Turned Pretty. which I'm kind of obsessed with. And so I thought it would be nice to tie in here. So in my tween years, when I was between 9 to 12 years old, I went to this deaf, hard of hearing sleepaway camp called Camp Sertoma. And I formed this core group of friends who had the same communication modalities that I did, signing and speaking. I have very, very fond memories of this camp. And it was truly my first immersion in deaf culture and community. Everything was done in sign language. All the camp counselors knew sign language, and it was just a really fun environment to be in. But unfortunately, those types of camps, they do have an age out limit. So once I aged out, I really missed having that ASL environment I could go to since I was in a mainstream school where only one other person knew sign language. So I looked for more opportunities during the summers, and I found the Minnesota Conservation Corps um, had an ASL crew where half the crew was deaf and the other half was hearing, but ASL would be the primary mode of communication. And this program was about a month long, and it was really hard work. We basically traveled around to different state parks. We attempted to restore hiking trails, stone steps, and there was a lot of invasive plant removal. This is basically the forest version of weeding out plants that were harmful to the environment. And the Conservation Corps was also somewhat of an exposure / introduction to environmental science. So while immersing myself in this ASL-rich environment, I started to become interested in environmentalism, forest preservation, and conservation. And since the Conservation Corps was only a one-time gig, you can only participate in it one time, I ended up applying for the Northwest Youth Corps, which was in Washington and Oregon. And this was a very similar program that brought my crew and I to different state parks to work. And the difference here is that the entire crew was deaf, hard of hearing, instead of being half and half. So this was also a wonderful experience that sort of made me more curious about environmental science. And then after that, I went to Explore Your Future Camp, or EYF at NTID when I was 16. And I remember being hesitant about RIT/NTID since it was so far from Minnesota. But this is what really sealed the deal for me in terms of imagining RIT/NTID as a place where I could find my path. I was interested in a lot of things, from environmentalism to healthcare to architecture. So I wasn't completely sure what I wanted to do, but I knew I wanted RIT to be in my future. Once I knew I wanted to go to RIT, I had to figure out how to pay for it. So my mother, when I was growing up, she always told me that she wouldn't be able to afford college for my sisters and I. So I knew that I'd have to find a way to pay for it. And a huge reason I could do the Conservation Corps, like these opportunities, is because they actually paid me for the work I did. And I also did a lot of bagging groceries, yard work, and retail jobs to save money for college. But once I graduated high school and became 18, I wanted to do something special for my last summer before college. So inspired by the Conservation Corps, I looked for opportunities that would let me travel, gain work experience, and then also get paid so I could save up money for college. This is when I ended up finding an opportunity called the Summer Student Program at the Jackson Laboratory in Bar Harbor, Maine. So this background is actually a picture that one of my friends in the program had taken of Maine, and it was like this really pretty picture. And this in particular was not a deaf program. I think at that age, when I was 17 or 18, the only deaf summer programs available to me were the ones that I couldn't afford to go to. So this was actually a mainstream program. It was a program intended to let students gain research experience in genetics while paying students a stipend, which, if you don't know yet, that's basically a lump sum of money paid once during your employment. And this program was targeted toward late stage high school students and college students. And at that time, I had absolutely no idea what doing research meant. It was a very vague idea and abstraction. But what I did know at the time was that, one, it paid a stipend. Two, I would get to live and adventure in Maine. And then three, I was somewhat interested in science, even if I wasn't sure what kind yet. At the time, I really only had exposure to environmental science, and this was definitely more the biomedical route. I figured it was a long shot because I had absolutely no research experience. But I ended up getting in, and it completely sparked my interest in biomedical science. And these are a couple of images from my project. My project focused on stereocilia on hair cells. So each of these little circles is a hair cell, and then all the spiky looking protrusions on it are stereocilia. They move around when sound goes into your ear because of all the different like pressure happening. And then this was just a plot that I ended up generating. If you're interested in science and you're going to stay in science, you're going to be making lots of plots like these. But you can also choose which colors to make them, which I think is very fun. And there were also things that we did outside of research. One of them was a program requirement that we had to hike Mount Katahdin, which is the highest peak in Maine. And so we spent a lot of time hiking smaller peaks before that to train for Mount Katahdin. And we ended up hiking Mount Katahdin at the end of the summer. And I think it was a very special experience. And actually now I think they've gotten rid of that requirement, which is a bit sad because I think it does do a lot for your character. It does take a while. We took about 10 hours to hike that mountain, but it was great. And the program also gave out a free book to everybody called The Gene. So this was something else I did outside of research. Someone told me that it would be impossible to finish it within the summer, and I took it as a dare. So I ended up reading the whole thing. I think it had about 600 pages. Mind you, the most knowledge I had was biology class that I took in high school. So it was a bit of a challenge to read it. But I'm pretty glad that I did because it started to introduce me to a lot of the language used to describe life sciences. And the lesson of the story is that even if you don't think you're qualified for opportunities you're interested in, go ahead and give it your best shot. You really never know what could happen. And as an aside, if any student is interested in exploring genetics research, I'd really encourage you to consider this program. The deadline is in January, and you can find the information on the Jackson Laboratory site. So when I came into RIT after finishing the summer student program at the Jackson Laboratory, I was a biology major. Well, actually, I was a biochemistry major on day one, and then on day two, I switched to biology. And that's a different story. But I was still kind of interested in environmentalism and biomedical science, and I figured biology would be the best major for me to narrow down those interests. And although I enjoyed a lot of classes at RIT, there were a couple of key classes early on that helped me ultimately narrow down my interest to biomedical science. and these were cell biology and biopsychology. So I ended up looking for research opportunities that were related to these areas. Over the course of my undergrad, I've been really lucky to have the chance to do research in a variety of different topics. I won't go over all of them here, but they all ultimately helped me gain the scientific skills and foundation that I needed for graduate school. What's important to note is that I could not have been able to do all of this without getting paid through all the funding resources that I had, which are listed here. And I especially want to point out RITU Rise, which was my primary funder throughout undergrad for most of these labs. And I want to talk about this training program for a minute. So many of the research opportunities I had access to, the access services I had, were because of the Undergraduate Research Initiative for Scientific Enhancement, or URISE. And from a financial standpoint, the URISE program was invaluable to alleviating the financial worries I had about paying for college while being able to focus on my education. It's a shame that with the current administration policy that this program has been closed. I hope that they will start it back up again, because I'm not sure that I would have been here today in the grad program that I'm in without you guys. No, that's going back. And if you noticed from this slide before, one lab is actually mentioned twice. So that's the Michelle lab. This is Dr. Leah Michel's lab in Gosnell College. I was actually in Leah's lab for all 4 years of my undergrad, so it has a really special place in my heart. And this is Leah. She was my mentor in undergrad. She really made science engaging and approachable. I remember when I came into RIT and was interested in biomedical science, I was actually very against doing a PhD. I wanted to gain work experience, find a job after graduation, and be done with school. I didn't want to be a student for longer than I had to. So even with the URISE program preparing students to become doctoral students, I definitely had my hesitations about it before joining. I was worried that if I committed to the program, I had to swear my life away to do a PhD. But Leah really believed in my potential. And I remember one time I was in her office telling her that maybe I'd do a master's at most. And she kind of looked at me like I had grown 2 heads and very seriously told me, I think you should do a PhD. And so I was like, okay, I'll think about it. And I'll explore the option within the URISE program to see if it's the right fit for me. And so I started to think about reason for doing a PhD or how it would help me attain my goals. And this was uncharted territory at the time because most of my family had never pursued graduate education at the time, and none of them had pursued doctoral training. And I also didn't know any other deaf scientists at the time. So it felt a bit like I was flying blind. But within this lab, which had other deaf students, and within URISE, I learned that I really liked scientific research and project management. So I started to see a PhD program as an important training step in my career, because often to reach project management roles in science, like research director, PhD is a required credential to have. In addition to Leah, the people in the lab were the reason I got to have the opportunity to speak at my college graduation. It's a nomination process by your peers, then by the college, and then by the president. So if it hadn't been for my peers believing in me, I wouldn't have had that opportunity. So the takeaway is remember that your peers are your first source of support. I know personally, I want to see all my peers succeed and thrive. So just remember that. And then once I graduated RIT, I knew that I was going to grad school, but I also knew I needed a break. So I took a gap year and I wanted to explore what life could be like if school was not in it. And what I didn't expect was I actually had a sort of mini crisis once I left school. It was all I had done for 13 years, and suddenly I didn't have to do it. I had become part of so many different communities at RIT that I ended up feeling a bit like this monkey, just sort of wondering who I was outside of these communities and outside of school. It's quite a shift to go from very structured days to suddenly long periods of unstructured ones. So what I did was I wandered a bit. I went to Japan, used the sign language Starbucks in Tokyo, Japan that I went to. I did a co-op at Merck with my lovely ID photo here. And I did this to experience science and industry. It's very different than academia and much more pharmaceutically focused. I caught up with friends and family, and I ran my first marathon. While I did feel like I was a bit in limbo during this year, I'm thankful that I got to sort of wander and reflect on things rather than just being go, go, go all the time. And I do want to point out that one really beneficial thing I did at Merck that I didn't realize at the time, I ended up taking the opportunity to test drive different interpreting agencies in Pennsylvania and making connections with the interpreters in the area. Um, I hadn't known that I was going to be going to grad school in Pennsylvania yet, but once I started grad school, I ended up requesting those interpreters that I previously worked with, which made the transition more seamless. And because I was pretty flexible on my journey this year, I ended up meeting some other really cool, amazing deaf scientists at the STEM Sign Language Conference. We bonded over our shared experiences of inaccessibility of STEM conferences, so we decided to make an article about it. And if you're interested in reading it later, this is the title, Illuminating the Deaf Experience at STEM Conferences. All right, so now into talking about grad school and my current research project. So here's a picture of what my studio apartment looked like when I moved in. These boxes were all I had. All my life possessions were in these boxes. And I ended up deciding on attending the University of Pennsylvania because I really wanted that city living experience. And then I also wanted to experience some of the incredible opportunities at UPenn focused on the intersection of business and research. My co-op stint at Merck really opened my eyes to how there's so many moving parts behind getting a drug developed and commercialized. So this is something I wanted to learn more about at Penn. I will say my first semester at Penn, there was some significant growing pains with getting access to services that I needed. It's much better now, but I did have to do a lot of advocating with individual people who had never worked with deaf students before, even if the institution itself has had deaf students. This is a pretty big story in itself. So if you're applying to graduate school and you're sort of wondering how to find a school or program that will provide the accommodations you need, feel free to reach out to me after this talk. All right, so my research project specifically focuses on this Cas9 CRISPR screening for glioblastoma myeloid cell infiltration. Don't worry if you don't understand what most of that means, because we will go over it together. So while I present this project, I sort of want you to think about the scientific process of understanding the background, the methods, results, and the next steps. And even if you don't fully understand, just sort of observe how we walk through the process, because that's something that can be applied to really any question that you have in your life. It doesn't have to be academia related. And so what are glioblastomas? Glioblastomas, shortened to GBM, they are this fast-growing cancerous tumor And they actually have, compared to other cancers, they have a very, very low survival rate. You can actually see in this bar plot here that the 5-year survival rate for glioblastoma is very low compared to the other brain tumors. And it's a very aggressive and incurable tumor. Currently, the way that you treat it is you treat it with radiation, chemotherapy, and surgery. And then there are different features or characteristics of these tumors. So one of the most important thing about these tumors is understanding the environment that it lives in. So the environment that it lives in is called the tumor microenvironment. And the tumor microenvironment itself is usually made up of a bunch of different types of cells. One of these specific cell types is myeloid-derived cells. And in each tumor, it's primarily composed of 30 to 50% of myeloid-derived cells. And what these do is, although it's not entirely clear how, what these cells do is that they will suppress your immune system. And they will suppress it in a way that your immune system cannot attack the tumor. And this lets the tumor grow and grow. And so that's why it's called pro-tumorigenic. And then another feature of it is that there are different molecular characteristics. So there's different mutations that happen in the promoter regions. If you've ever heard of like an oncogene, that's a gene that's especially prevalent in cancer. So like EGFR is an oncogene, and in glioblastomas, EGFR is amplified. So there's extra copies of this. And because there's extra copies of this, the oncogene is going to be more active, and it's going to help the tumor grow. And then where it's typically located, these tumors, it's in the cerebral hemispheres, particularly the frontal, which is what you can see here, and either the temporal lobe as well. And they can also be divided into different subtypes just for diagnostic purposes. And one of the things in the research process of understanding what we know and what we can learn is you want to understand what are the research gaps in the area. So in this particular case, there's a huge lack of knowledge about these myeloid-derived cells. And what we do know so far is that these myeloid-derived cells, they come from these hematopoietic stem cells. And these hematopoietic stem cells, they come from your bone marrow. So these myeloid cells, they can differentiate and give rise to many other different cell types. But we're really interested in seeing how they specifically lead to myeloid-induced immunosuppression. So how do they suppress the immune response? And the general consensus, or as much as we know, is that the tumor cells, along with some other cells, will release signals that are called growth factors or inflammatory cytokines. And these signals will go to the bone marrow, and they will tell the bone marrow, okay, I want you to produce XYZ. And in this case, the XYZ is MDSCs, which are just a type of myeloid-derived cell. And these cells will go back up to the tumor in the brain, and they will inhibit the immune response. So this brings us to the main question of what myeloid intrinsic genes and pathways regulate myeloid infiltration in GBM. So how can myeloid cells infiltrate these glioblastoma tumors? And this brings us to a few different hypotheses. The first of which is that genes involved in chemoattraction axes will serve as facilitators of myeloid infiltration in glioblastomas. And chemoattraction basically means a specific ligand will come to a cell and they will induce this signaling process that tells the cell what to do. And in this case, the hypothesis is that the tumor cell will release a ligand that travels to the myeloid cell and tells it to come and infiltrate the tumor. And then the second hypothesis is that genes involved in phosphatase mediation of immune checkpoints, such as PD-L1, serve as facilitators of myeloid infiltration and glioblastomas. And what this means is that a lot of times our immune system will activate certain cells called T cells to attack tumors. But sometimes tumors and maybe myeloid cells will produce PD-L1. And what PD-L1 does, it basically stops the T cell from attacking by hiding the cell. So PD-L1 will allow the cell to escape from the T cell. And then the third hypothesis is that genes encoding for transcription factors that limit expression of cytotoxic mediators could serve as endogenous brakes for myeloid infiltration. The first two hypotheses are about which genes help infiltration of myeloid cells in tumors. And then the third hypothesis is about which genes actually help stop or prevent infiltration in tumors. And moving on to the methods of how can we answer these questions to support these hypotheses. What I am doing currently is a in vivo, so that means in a living system, perturbation CRISPR screen. And what that essentially means is that I will disturb or knock out a bunch of genes at once. And then I'll see which cells will go up into the tumor in order to see which genes help infiltration and which genes do not help infiltration. And the general process is that I harvest these hematopoietic stem cells. If you remember earlier, these are what give rise to myeloid cells. And I'll take these and then I'll put in all of these different what's called guide RNAs. These guide RNAs are what cause the gene knockouts. So what ends up happening is I have this pool of different cells, and each cell has a different gene knockout. And then once I have that ready, I end up injecting them into these mice in order to reconstitute the immune system. So previously, mice, they already have their immune system ready. But what I need to do for my project is I need to give them donor immune cells and see where they go. So what I have to do is I have to actually eradicate the existing immune system in mice before I can give them donor immune cells. And then once that happens, after a while, as I wait for them to be fully replenished with donor immune cells, I'll end up injecting these tumors into their brains. So this is the tumor condition, and then this is the no tumor condition, and then this is just another type of control. But eventually what I want to happen is the tumor itself will select for the cells that have specific genes that help or inhibit infiltration. And then I take these cells out, I harvest them, I analyze and sequence the DNA. And so going back to this hypothesis page, I want to start thinking about what are the expected results. So for the first hypothesis, the expected result is if I compare the tumor versus the control condition, is that there's going to be a decrease of cells that have gene knockouts. And then it's the same for hypothesis 2, is that there will be a decrease in cells that have gene knockouts in the tumor compared to the control condition. And it's the opposite for the third hypothesis. I would expect an increase in gene knockouts compared to the control condition. And once I get my results from the sequencer, I have to put it through this program. It's called Castle-CRISPR Screen Analysis Pipeline. The main idea is that what it will do is it will take in all of the data from the sequencer, and it will analyze it and produce output plots that will give me meaningful results. And so this is one example of an output plot. Before I actually start with the analysis of the entire data, I do have to make sure I do quality control. That's what QC means, quality control checks, just to make sure that the data is behaving the way that it should. Because if it doesn't behave the way that it should, then that means you can introduce technical variability into your data, and you end up getting a result that's not true. And that's essentially what these quality control checks do, is they just check to make sure that all the single guide RNAs, all the gene knockouts are behaving the way that they should. And this normal distribution is what I would expect to see for each plot. After doing the QC checks is when you have to analyze for the actual results. And also, as a disclaimer, all the results that I show within this presentation is based on simulated data. I haven't analyzed my acquired data yet because I just got it on Friday, which is very exciting. But this is all just kind of a general schematic of what I would expect to see. And this plot does still need some work, but basically what it's showing is that of all the genes that I knocked out, what is the significance of the gene and where is it located? So like, for example, the chemo attraction genes, which are labeled chemo genes, I would expect to see these more in the top left quadrant because I would expect them to be upregulated and I would expect them to be significant. So, and then for the immune mediation genes, I would also expect these to be in the top left quadrant. For the breaks, they would be on the opposite side, so on the bottom right quadrant. As you can see, the examples listed here don't entirely follow that to the letter. That's because I still need to fix the simulation. But this is an example of an output plot that would provide a meaningful result. And then here's another output plot. It's called a volcano plot because it looks like a volcano has gone up. Each dot is a gene, and it shows the comparison of tumor versus no tumor. And what it shows is that these genes that are in red and they're on the right, these are significantly upregulated compared to the baseline. And these genes on the left, which are downregulated and significant, they are also representative of maybe the breaks. And then another output is this GC analysis, which stands for Gene Set Enrichment Analysis. And what this does is basically there are a bunch of genes that people already know are involved in specific processes. And so we take those genes, those groups of genes, like chemokine-mediated signaling pathway, and we look at our own data set and ask how many times did that gene pop up in our own data set? And does it pop up in excessive amount compared to the control? And so if my hypotheses prove correct, then all the groups of genes involved with chemokine-mediated signaling and response to chemokine's the ligand, and chemoattraction, which should be upregulated in tumors compared to the control, because they will help facilitate myeloid cell infiltration. And then for negative regulation of cytokine production, this is like the group of genes that could potentially serve as brakes and inhibit myeloid cell infiltration. And then this is just an individual plot that's focusing on like one of those pathways. And it looks at every single gene to see the levels of that gene expression. But this is the chemoattraction pathway plot. And essentially, if the peak is on the left side, you'll see enrichment and upregulation. But if the peak is on the right side, then it will just provide support against the hypothesis. And then these are just some of the next steps that I need to do for my project. The most exciting thing is analyzing those initial results that I told you I got last Friday. I just haven't had time to analyze them, but these are my first very real results in my project. So I'm excited to see what I get. And I just wanted to give a thank you to everyone who helped me along the way, my mentor, my current lab, different summer programs. And I also played on the Ultimate Frisbee team, so that's what you see here. And then my best friends, Amy and Sophia. This is when we were on a study break at RIT, and we really wanted to just enjoy the weather in Rochester. So thank you.